What we call in scientific, non-scientific jargon is that we call them reader and writer. So writers are the protein molecule or the enzymes. They write on the DNA, write on the histone molecule, and readers are some of the guys. They will look at, oh, this is a place where DNA is methylated or histone is methylated. Then these proteins have specific domain like PSD domain or chromo domain. They recognize these signatures on histone or DNA methylation. They will sit down there and make a much stronger repressor complex. It is like, imagine a situation it's like an ant colony. They find sugar, one ant senses, and that ant's sense will invite the other ants to come together, and after some time the big ant gathering will happen. In the same way, if you have a methylation signature, first the methyl binding protein will look into that and bind, then the chromatin will have other factors that are going to bind onto the methyl binding protein, and they make a transcriptionally impossible complex. Now the question now comes is that, okay, if a gene is transcriptionally silent, how does it get unsilent basically, or how do you revert back? So for that, actually many a time you will see that it could be a complete change of signature on the chromatin. Especially you will see histone deacetylation is transcriptionally silencing, but histone acetylation is transcriptionally active. So the histones which are transcriptionally silent, now they get acetylated. So there is a series of proteins again called histone acetyltransferase. They will come and take care of the business. In the same way, DNA demethylation can occur. Now recently we know that 5-methylcytosine can be converted to 5-hydroxymethylcytosine, which is an oxidative product of the signature that was involved in gene silencing. But more and more knowledge is coming that 5-hydroxymethylcytosine probably is a transcriptionally active signature, either via a repair mechanism where 5-hydroxymethylcytosine get removed and cytosine comes into the DNA strand by erasing the signature and making the gene active, or simply that 5-hydroxymethylcytosine does not allow those proteins to attach to the repressor complex, thereby just keeping the track clear. So the protein molecule can go down the track. mRNA will go down the track and make the protein. Other than the methylation signature and acetylation signatures on DNA and histone molecules, there are other small molecules also involved in epigenetic processes, such as microRNA, small RNA, sometimes even long non-coding RNA. So these are involved in also modulating how a gene should work and what time frame it should work, and what is the environmental factor it is going to respond and work. So if you look into epigenetics, it's a very big field, and there are multiple players here, and it's a very complex field. So epigenetics is essentially a very important phenomenon. It participates in development, diseases, and many other kind of neurological, psychological things too.